Hi, I'm Rebecca Dikow. I'm the Biodiversity Genomics postdoc. So half of my postdoc is developing collaborations with scientists across the Smithsonian, particularly at the museum here and at the National Zoo. The other half is helping with the capacity building in terms of biogenomics in general, in terms of partnerships with Oak Ridge and bioinformatics pipelines. But today I'm going to talk to you about the third half, which is extension of my dissertation research, which is what I'm really interested in, which is using whole genomes to develop phylogenetic hypotheses. So here I'd like to talk about using genomes across all three domains of life to find really deep homologies. And so here are four hypotheses of how these three groups could be related. So bacteria and archaea. Archaea were initially thought to be part of bacteria. They're both prokaryotes, they have no nuclei, they have very similar structures. But in the top right, you'll see the first phylogenetic analysis that was done across all these three groups using ribosomal RNA showed that maybe archaea are more closely related to eukaryotes. In the bottom right, you'll see a more recent hypothesis that eukaryotes are nested within archaea, and this was done using only coding presence and absence of genes. You can also imagine that the base is really hard to resolve here, so maybe it's a trichotomy, and also maybe none of these groups are monophyletic. So what I wanted to do was look at genomes across all these things. So previous hypotheses consider only a few genes, or simply code presence and absence of genes, but we're talking about a 4 billion year divergence. So while function of genes may be sometimes a proxy for homology, it may not be 4 billion years ago. You can imagine a lot of convergence has happened in function. So here, I'm looking at complete genomes. I'm ignoring annotation. Annotation is the assignment of genes to parts of the genome, ignoring gene boundaries and traditional assumptions about what is homologous. So this is what I've done. I've downloaded all the genomes on GenBank. It's 2,000 species. I've only included the finished bacteria and archaea. By finished, I mean a genome that is at the chromosomal level, so each contig is assigned to a chromosome. These are closed genomes. For eukaryotes, in the interest of including more than just a couple, I've included draft genomes as well. So these are sets of contigs or scaffolds. I've done pairwise alignments with LASZ, which is a genome aligner. So this is pairwise. There is no way to do a global heuristic version of this for this many species, although developing that kind of method is of utmost interest to me. So I've chosen a target that is bacteria, and I don't have time to talk about why and the reasons for that, which I can later if anyone has questions. It's 4.5 million base pairs. And so on the bottom you see the query is either a set of chromosomes for eukaryotes, a large number of chromosomes, or up to, even in some cases, hundreds of thousands of contigs that exist for these genomes. And LASZ works, is based on BLAST, but it finds parts of these contigs or chromosomes that match the target. And then once you do this over and over, so you have the target, complete genome, and then you have the parts of all the different queries that match up, and then you get really just, it's a huge alignment. So this is the first tree. So I was able to run a single maximum likelihood tree in a couple of weeks, and this is brand new data. So this is a tree of pretty much everything. So what I'd like to point out is that bacteria are not monophyletic, but what's interesting is that the phyla are. So I was happy to see that. There are about 1,500 bacteria and archaea species here, and about something like 500 eukaryotes. So archaea are monophyletic, but you can see eukaryotes are not. I think I was surprised by the number of fungal genomes there were, and so it's not monophyletic, but there are only 6 that are outliers. So you'll see there are some strange things. So trypanosomes and brown algae, it's really, I don't really know necessarily where those should go. There are very few protist genomes, so this is a problem. I think this tree helps point out where are the problem areas. So protists is a big one. There were a few protists that I had to remove that were draft genomes An interesting thing about draft genomes is they often have bacteria in them. They have whole contigs that, in many cases, that was the only thing that matched. So I threw those genomes out. I could go back and in a fine-tuned way, clean up those genomes, but I think maybe that's for those genome projects to work on as well. Diatoms, again, they don't group with cyanobacteria on plants. And the non-insect invertebrate clade is, it has a lot of singleton taxa in there, and the non-insect arthropods are grouping in there as well. But I had to remove a lot of those. For example, the tick was showing was bringing an entire clade of bacteria into the invertebrate lineage. So rickettsia is the pathogen that ticks carry, and so it brought the whole rickettsia clade into there. And when I looked at all the matches for the tick genome, they were all bacteria. But I think this is really interesting, and it points out some of the challenges that come with draft genomes, but they're still useful. So there are very few eukaryote genomes that are finished, but we can still use about 500 of them to, I think, some interesting results. So finally, this is the same kind of cartoon. There's no timing on this tree, so we imagine all these splits to be billions of years old. But eukaryotes are not monophyletic, as this is a single search, so this is not a final result. And Archaea seem to be nested within bacteria, but I'm happy to take questions.